Hello everyone, my name is Kristen Edwards and I am going to be presenting my genetic disorder, beta thalassemia. Alright, so I wanted to do a quick little review on red blood cells. This here would be a red blood cell. Inside each red blood cell there's hemoglobins, and this hemoglobin is zoomed in over here. You would have 2 alpha globins and 2 beta globins. So, and then each of these globins from the heterotetramer hemoglobin has a iron attached, and this iron, or the heme group, which has the iron, which allows for oxygen to be attached, and this is how oxygen gets from your blood and into your other tissues. So the hemoglobin beta gene is the one involved in making the instructions for this subunit right here, this beta globin. All right. So I'm going to do a quick overview on beta thalassemia. So the body reacts by destroying red blood cells, causing anemia. So here you would see somebody with anemia having a lot less red blood cells available compared to somebody who has normal blood. The body tries to compensate for the loss, which causes other thalassemia complications such as splenomegaly, which is an enlarged spleen, or this sort of chipmunk face, which is from bone marrow abnormalities. So without enough beta globin, there will in turn not be enough red blood cells in the body to distribute oxygen to the rest of the body. Right. So here we have our affected population, and if you can see, everything in orange is the thalassemia. So there is 2 types, actually, beta and alpha thalassemia. However, we are focusing on beta. Either way, beta thalassemia affects people in the Mediterranean, Middle East, Africa, Central Asia, and Indian subcontinent. It's relatively rare in the United States, but it is actually one of the world's most common autosomal recessive disorder, occurring within 1 in 100,000 individuals. So as I just said, it's autosomal recessive. So here you have your autosomes. It's from chromosomes 1 to 22. The sex chromosomes would be the 23rd chromosome. The HBB gene, as you see here, is on this chromosome 11. So right here is our chromosome 11s. It must be inherited by both parents, hence the recessive. And there are several mutations that cause beta thalassemia. Most often are said to be substitution of one nucleotide, an insertion, or very rarely a deletion. There are 3 subclasses of beta thalassemia. You have beta thalassemia minor, intermediate, and major. So with beta thalassemia major, there is 1 mutation in the HBB gene, and they are also considered carriers for the disorder and are many times asymptomatic, which they could also have very mild anemia, which would not really cause them to have too many issues. All right, so then we have beta thalassemia intermedia and beta thalassemia major. So with intermedia, you have moderate to severe anemia. You can show pallor or jaundice, can have some leg ulcers and gallstones. And also this is a mutation that's seen in both genes. So let's see. Yep, so then you have your, the beta thalassemia major is usually diagnosed within the first 3 to 6 months of life, and the babies tend to be very pale or even jaundice. It's a thalassemia. Beta thalassemia is a lifelong chronic illness with no known true treatments, only just to help the effects of it. So with anemia, someone with a beta thalassemia major might have some anemia, which would make them weak, shortness of breath, dizzy, or have headaches, and also that splenomegaly, so that enlarged spleen. which might show by an enlarged belly as the spleen becomes overactive with the buildup of blood cells that are destroyed in the spleen. Okay, and then our bone marrow, like we saw in the chipmunk face, is in response to that anemia, and it makes the bones very weak and thin. So I thought this was a very cool pedigree. It doesn't just show if somebody has the trait, or has the genetic disorder or does not have the genetic disorder. It kind of shows the different types here. So for example, this individual right here has thalassemia major. Their sister also has thalassemia major, and they came from parents who were a silent carrier and who had beta thalassemia trait. So I thought this was cool. Little pedigree I found online. As far as diagnosis, a person who is thought to have beta thalassemia will get a CBC, so a complete blood count. It's going to measure the number, concentration, size, shape, and maturity of blood cells. An abnormal result would have fewer red blood cells, smaller red blood cells, an increase in iron, decreased amounts of HbA, and increased amounts of hemoglobin F. This CBC could also be in turn turned into a blood smear. So that's, they drew the blood here, they got their CBC, and they took part of it to do a blood smear. This would be a microscopic view of beta thalassemia, where these individuals have microcytic, very small red blood cells, hypochromic, so hypochromatic cells, which are pale in color, and they might have what they call a bullseye cell, which is that target cell, which is labeled right here. So here's another one. It kind of looks like a bullseye. So this is usually done initially with a newborn heel prick, but that does not give any sort of confirmation. Further testing would have to be done later on in life in order to diagnose beta thalassemia. All right, so as we said before, we usually use a CBC and a blood smear, but there's very rarely we use genetic testing. It's not usually a standard practice unless there is a risk for a relative. So for example, I have it, so my sister might get tested. And also prenatal screening. So the prenatal screening can be done during an amniocentesis. So that's this picture right here, where a sample of the fluid surrounding the fetus, so this fluid right here, would be tested, or a chorionic villus sampling, so a CVS. It's removal of the tissue samples from a portion of the placenta. So that would be this type of, and then that would, once those are collected, they would be tested genetically using maybe a quantitative PCR, multiplex ligation dependent probe amplification, and chromosomal microarray. So you would identify biallelic pathogenic variants on the HBB for molecular testing. So here is that heel prick we talked about, that initial testing on the babies. So as we said before, it is a chronic illness, and some people might be prescribed some folic acid supplements to help with their mild anemia. This will boost the production of blood cells. Blood transfusions are also used for most intermediate and beta thalassemia major cases. And in this case, you have to be very careful with the buildup of iron. This buildup of iron could possibly lead to cardiac dysfunction, arrhythmia, liver injuries, and so forth. And you might also get that splenectomy because of your splenomegaly, which just means the enlargement of your spleen. And that can cause issues with low levels of platelets, and those platelets usually allow the blood to clot. Also, extra hemolysis, so that's the rupture or destruction of your red blood cells. And we have our resources for this presentation here. And thank you so much. You guys have a great afternoon and the rest of your time here in human genetics.